Good morning, all. Good morning, students. In the last lecture, we were discussing about the hybridization methods in the self-pollinated crop. Okay, we have discussed that the 1 minute. We have discussed that the handling of F1 generation and the further segregating generation. Okay, that is F2, F3, F4 generation. It is completely different for the self-pollinated crops and the cross-pollinated crop. Okay, handling of F1 generation and the subsequent generation, it is different for the self-pollinated and the cross-pollinated crop. Okay, and we were discussing the hybridization methods in the self-pollinated crops. There are four methods which are used in the self-pollinated crop. The first one is the pedigree method, second one is the bulk method, third one is the backcross method, and the fourth one is the multiple or the composite cross. Okay, in the last lecture we have discussed the two methods of these four methods in detail. Okay, we have discussed the first method, pedigree method, and the second one, bulk method, in detail in the last lecture. Okay, in the theory examination the question may be asked, long answer question may be asked as what is mean by hybridization, and give a detailed account on hybridization methods in the self-pollinated crop. Okay, there are four methods which are used for the hybridization in the self-pollinated crop. The first one is the pedigree method, second one is the bulk method, third one is the backcross method, and the fourth one is the multiple or the composite cross, of which two methods, pedigree method and bulk method, we have discussed in detail in the last lecture. And in today's lecture we are going to discuss the remaining two methods, backcross method and the multiple or the composite cross. Okay, here you can see. Okay, backcross method. Backcross method, it is the method which was firstly proposed by the Harlan and Pop in the 1922. Okay, backcross method, it is proposed, it was proposed for the first time by the Harlan and Pop in the 1922. The backcross method, it is entirely different from the pedigree method and the bulk method. It is the method which can be used, which can be employed in the improvement of both the self and the cross-pollinated crops, where the varieties are deficient in one or two aspects or one or two characters. Okay, this method, it is completely different from pedigree and the bulk method. This method was first proposed by the Harlan and Pop in the 1922. This is the method which can be used in both self and cross-pollinated crops where the varieties are deficient in the one or more characters. Okay, this method, it is used particularly for transferring a single simple inherited characters, single simply inherited characters like disease resistance, frost resistance, or the drought resistance and earliness from an undesirable variety to a good commercial variety. Okay, this method, it is used particularly for transferring a single simply inherited character. Single simply inherited character, for example, disease resistance is there, then frost resistance or drought resistance and earliness from an undesirable variety to a good commercial variety. Okay, here the desirable variety, it is called as the recurrent or the recipient parent. Okay, the desirable variety, it is referred as the recurrent or the recipient parent. Okay, recurrent or the recipient, and this desirable variety, or we can say the commercial variety, it is crossed with an undesirable variety. Okay, and that undesirable variety, it is called as the donor or non-recurring parent. Okay, donor or the non-recurring parent, because this parent is called as the donor because the desirable genes, or in other words we can say the desirable characters, are transferred from this undesirable variety into the desirable or the commercial variety. Okay, do not forget that the desirable variety, or we can say the commercial variety, in which the character are to be transferred, this variety, it is receiving the character from the another undesirable variety. Okay, and that's why the desirable variety or the commercial variety is called as the recurrent or the recipient parent. Okay, and this commercial or the desirable variety, it is crossed with an undesirable variety, and undesirable variety is called as the non-recurring parent, and also it is referred as the donor parent. Donor parent because the character or the desirable character, it is to be transferred from this parent into the desirable or the commercial variety. Okay, for example, there is a variety which is good in all the characters, but it is not a disease resistance. It is not disease resistant, it is susceptible to the different diseases. That variety, it is said to be the desirable variety, or it is the commercial variety, it is the recurrent or the recipient variety. And another variety, another parent is the same variety, but it is a growing as a wild species, but it is having the disease resistant character, but it is very poor in remaining character. Okay, and from that variety, from that wild variety, the disease resistance is to transfer into the commercial variety. Okay, and therefore the wild variety, it is said to be the undesirable variety, and as it is transferring the disease resistance into the commercial variety, therefore this wild variety is called as the donor variety or the non-recurring parent. Okay, it is said to be the non-recurring parent. Okay, non-recurring parent. Okay, here then next, here you can see here the F1 plants or the F1 progenies. Instead of selfing, these are, instead of permitting the self-pollination as in pedigree or the bulk method, these F1 progenies, these are crossed with the recurrent parent. Okay, the F1 progeny, instead of permitting the self-pollination as in pedigree or the bulk method. Okay, we have discussed the pedigree and the bulk method. In this method, the F1 progenies, these are allowed for the self-pollination. Okay, but in the backcross method, the F1 progenies, instead of permitting the self-pollination as in pedigree or the bulk method, these are crossed with the recurrent parent. Okay, these are crossed with the recurrent parent, and therefore this method is called as the backcross method. Okay, the objective of the backcross method is to recover the genotype of the recurrent parent with the all the good characters. Okay, the objective of the backcross method or objective of crossing the F1 progenies with the recurrent parent is to recover the good characters of the recurrent parent. Okay, the progenies which are produced due to this backcross, these are called as the backcross generation 1. Okay, here you can see these are called as the BC1. Okay, the progenies which are produced by crossing the F1 progenies with the recurring parent are said to be the BC1. Okay, backcross generation 1. Okay, backcross generation 1. Okay, then from this backcross generation 1 or the BC1, the progenies, having the good characters from the donor plant, these are crossed again with the recurrent parent. Okay, from backcross generation 1, the progenies which are having the good character from the donor plant. Okay, in this case we can say the donor plant, it is donating the disease resistance capacity. Okay, that means progenies with the disease resistance capacity, these are again crossed with the recurrent parent. Okay, progenies having the good characters from the donor plant in backcross generation 1, these are again crossed with the recurrent parent. Okay, these are crossed again with the recurrent parent, that is to recover the all the genotype, all the genes, all the good characters of the recurrent parent, okay, or the recipient parent. Okay, and that process, it is continued up to the backcross generation 5 or the backcross generation 6. Okay, the process is continued up to the generation 5 or the sixth generation. Okay, in each of the generation, the plants possessing the desirable characters of the non-recurrent parent, okay, or we can say the undesirable variety. Okay, don't forget that in each generation, the plants possessing the desirable characters of the non-recurrent parents or the undesirable variety are primarily selected, and these are crossed with the recurrent, or we can say the commercial variety. Okay, always remember that in each of the generation, the plants possessing the desirable characters of the non-recurrent parent or undesirable variety are primarily selected, and these are crossed with the recurrent or the commercial variety. Okay, and it is continued up to the fifth or the sixth generation. Okay, and at the end of the sixth generation, what happened? The selected plants, these are allowed for the self pollination. Okay, at the sixth generation, the selected plants are allowed for the self pollination to make the population homozygous in the combined characters. Okay, at the end of backcross generation 1, backcross generation 6, the selected plants, these are allowed for the self pollination to make the population homozygous in combined characters. Okay, here in this type of cross, if the selection is made properly, if the selection is made properly, the plants which are obtained at the end must closely match the original parent, except differing from it only in desired gene incorporated from the donor plant. Okay, if the selection is made properly, the plants obtained at the end must very closely match with the original parent, except differing from it only in desired gene incorporated from the donor plant. Okay, this method, backcross method, it is called as the introgressive hybridization. Okay, this method, it is also called as the introgressive hybridization. Okay, this is the technique which is used in the backcross method. Okay, with the help of the photograph, here you can easily understand the method of the backcross. Okay, how the backcross method, it is used for the improvement in the self pollinated crop. Okay, in the first year, here you can see there are two parents here, the recurrent parent, which is here referred as the A. Okay, recurrent parent, or we can say the commercial variety or the good variety. That means this A variety, recurrent parent, it is having all the good character, but it lacks the disease resistance. Okay, suppose here we are taking the character which is to be transferred from the donor plant into the recipient parent is the disease resistance. Okay, here is the example. Here you can see there are two parents, A parent and B parent. Here A parent is the recurrent parent or the recipient parent, or we can say it is the desirable variety or the commercial variety. Okay, desirable or good commercial variety, which is having all the good character, but it lacks one important character, and this is the disease resistance. That means that variety it is disease susceptible. Okay, but here the another parent is used, it is referred as the B. Okay, here it is referred as the B parent. It is a non-recurrent parent. Non-recurrent parent, or simply we can say it is a undesirable parent, or it is a undesirable parent. Why it is undesirable? Be- because it lacks all the good characters. It lacks all the good characters, but it is having only a single most important character, is that this B parent it is having the disease resistance capacity. Okay, it is having only one good character, which is disease resistance, which lacks in the A variety. Okay, which lacks in the A variety. And in this program, in the backcross, we have to transfer, or the plant breeder have to transfer the disease resistance of this non-recurrent variety or undesirable variety into the good commercial variety, which is here referred as the A variety. Okay, as this non-recurrent or the undesirable variety, it is transferring the disease resistance from B variety into the A variety. That's why this non-recurrent or undesirable variety, it is also referred as the donor plant. Okay, it is donating its good character, okay, its disease resistance capacity to the A parent. That's why the A parent here, it is also referred as the recipient parent, and here the B parent, it is referred as the donor parent. Okay, here in the second year, here you can see the F1 progenies which are produced. Here the F1 progenies which are produced from these progeny, the progenies having the progenies having the disease resistance capacity of the non-recurrent parent B, these are selected. Okay, here the different types of progeny will be produced in the F1 generation, but only the progenies which are having the disease resistance capacity from non-recurrent parent B, these are crossed with the recurrent parent A. Okay, and this method is called as the backcross method. Okay, I'm repeating this one. The different types of the progenies will be produced in the F1 generation, but the progenies having the disease resistance capacity of the non-recurrent parent B, these are crossed again with the recurrent parent A, and this is called as the backcross method. Okay, and the progenies which are produced from this generation, these are called as the backcross generation 1. Okay, these are called as the backcross generation 1. Okay, from this generation also, the progenies having the disease resistance capacity of the non-recurrent parent, these are selected, and these are again crossed with the recurrent parent A. Okay, the process it is continue up to the sixth generation. Okay, up to the sixth generation, that is the progenies having the disease resistance capacity of the non-recurrent parent B, these are selected, and these are again crossed with the recurrent parent A. Okay, up to the sixth generation. Okay, it is mainly to recover all the good characters from the recurrent parent. Okay, and at the end of the sixth generation, then these progenies, these are then allowed for the self pollination. Okay, at the end of sixth generation, these are not crossed with the recurrent parent, but these are allowed for the six, these are allowed for the self generation. these are allowed for the self pollination. Okay, these are allowed for the self pollination. For 2 to 3 generation, the self pollination is carried out in these progenies, and that's why these progenies will become homozygous in all the good characters. Okay, these are subjected for the self pollination for 2 to 3 generations, so that the progenies with the all desirable characters, or progenies with all combined characters, these become homozygous. Okay, and then after their preliminary yield trials, this variety it is then with released and the seeds of these variety, these are multiplied, and then these seeds are provided to the farmers for the commercial cultivation. Okay, here you can see the variety which is produced, or the progenies which are produced at the end. They will have these progenies, these will be similar to the recurrent parent in each and every character, but it is differing from this recurrent parent only in the disease resistance. Okay, that means the variety which are produced at the end, they will closely match with the recurrent parent only in that character which is transferred from the non-recurrent parent B. That means the variety which is produced at the end, it will have all the good characters of the recurrent parent A and the disease resistance capacity of the non-recurrent parent B. Okay, understand that? Here, the another important thing in the backcross is that when here we have discussed the transfer of only one character from the undesirable variety into the desirable variety. Okay, but when the two characters of the donor variety have to be transferred, it is advisable to transfer each character in the separate program. Okay, in this slide we have discussed the transfer of only one character, and that is the disease resistance character from the non-recurrent variety B into the recurrent variety A. Okay, but when sometimes the plant breeder have to transfer the more than one character, suppose he wish to transfer the two characters from the donor plant into the recipient parent, then it is always advisable to transfer each character in the separate program. Okay, then this type of transference of genes of one species into the genotype of desirable species with the good characters by crossing is known as the introgressive hybridization. Okay, this type of transference of genes, okay, this type of transference of genes of one species into the genotype of desirable species with good characters by crossing is known as the introgressive hybridization. Okay, this is the technique which is used in the backcross method. Okay, then the next one, the advantages. What are the advantages of the using the backcross method? Okay, in this method the role of environment is almost negligible. Okay, role of environment, it is almost negligible. This method it is independent of environment. That means it can be used, this method can be used in any environment where the plants can grow and where the characters under transfer will express itself. Okay, therefore the role of environment is almost negligible in the backcross method, because this method it is independent of environment. That means this method can be used in any type of environment where the plants can grow and the character under transfer will express itself. Okay, then the second advantage is that the extensive yield tests are not required. Okay, extensive yield tests are not required because the performance of the recurrent parent, the performance of the recurrent parent or the recipient parent or the good variety or the commercial variety, it is already known. Okay, that's why the extensive yield trials are not needed. Okay, in other word, we can say there is no need of evaluation of agronomic performance. Okay, that is, the variety released by using this method can be safely distributed to the growers without evaluation of yield, adaptation, or the quality. Okay, because the recurrent parent or the commercial variety, it is already well known and the established variety. Okay, there is no need of evaluation of the agronomic performance, that is, the variety released by the backcross can be safely distributed to the growers or to the farmers without evaluation of yield, adaptation, or the quality. Okay, then this method is a rapid process. This method is a rapid method. Then this method, in this method, the small number of the plants are required. Okay, this method requires the small number of plants. Okay, it is not needed, or it is not required to select the large number of the plant. Okay, this method, it requires small number of the plants. Then another advantage is that this method is predictable. This method is predictable, and the use of this method have the greatest advantage in producing mainly the disease resistance variety. Okay, the large number of the disease resistance varieties are released by using the back cross method. Okay, the greatest advantage of using this method is in producing the disease resistance variety. Okay, these are the advantages of the back cross method. Okay, back cross method, it is entirely different from the pedigree method and the bulk method. Okay, and this is the method which can be used in both self pollinated and cross pollinated crops. Okay, though there are certain advantages of the using the backcross method for the improvement in the self pollinated crop, there is also certain limitations to this method. Okay, now the limitations of the backcross method. Okay, limitations: the new variety is superior over the already available variety only in the transferred character. Okay, the new variety which is released, it is superior over the already available variety only in the transferred character. Okay, then the another limitation is that the hybridization has to be done for each back cross. It requires the lot of time and money. Okay, the hybridization has to be done for each back cross, and it requires the lot of time and money. Then it is sometimes the undesirable genes, these are closely linked with the desirable one, and these undesirable genes get transferred to the new variety along with the desirable genes. Okay, here we can see the sometimes the undesirable genes are closely linked with the desirable genes, and these undesirable genes also get transferred along with the desirable genes into the new variety. Okay, then the another important drawback of this method, it does not permit the achievement of unusual combination of genes from the two or more varieties. Okay, it does not permit the achievement of unusual combination of genes from the two or more varieties, though there are certain limitations for using the backcross method in the self pollinated crop, but this method has the greatest advantage in producing the disease resistance new variety. Okay, the new varieties with the disease resistance, these are released by using the backcross method. Okay, that is all about the backcross method. Okay, the short note may be asked in the examination on the backcross method. Okay, then the another method which is used in the self-pollinated crop is the multiple or the composite cross method. Okay, multiple or the composite cross method. Okay, it is the method. The multiple or composite cross means it consists of crossing of several pure lines together. Okay, it consists of crossing of several pure lines together. Okay, generally this method it is used to combine the monogenetic characters from the different sources into the single genotype. Okay, this method it is used to combine monogenic characters from the different sources into the single genotype. Okay, suppose here the selected pure lines are first combined. Suppose there are large number of the pure lines, and from all these pure lines the characters are to be combined together into the single variety. Suppose there are several pure lines like the A, B, C, D, E, F, G, H. Okay, these are the large number of the pure lines, and from all these pure lines, all the desirable characters are to be transferred into the single variety. Then, in that case, this multiple or the composite cross method is used. Here, first of all, the selected pure lines are first combined into the single crosses as A into B, C into D, E into F, G into H, and so on. Okay, the first of all, these selected pure lines are first combined into the single crosses as A into B, C into D, E into F, G into H, and so on. Okay, and the F1 progenies of these single crosses, these are mated together. Okay, that means the progeny of A into B, it is crossed with the progeny which are produced by crossing the C and the D pure line. Okay, and just like that, the progenies of the cross E into F, these are crossed with the progenies of G and H. Okay, and finally the F1 plants of the double crosses. Finally, the F1 plants of these crosses, these are crossed with each other to produce the hybrid variety. Okay, finally the F1 progenies of the double crosses, these are crossed with each other to produce the hybrid variety. Okay, this cross it is also known as the multiple cross, and the further breeding in these hybrids it is carried out by either pedigree or the bulk method. Okay, after producing the hybrid, these hybrids, these further breeding in these hybrids it is carried out by the either pedigree or the bulk method. Okay, pedigree or the bulk method. Okay, further breeding in these hybrids it is carried out by the either pedigree or the bulk method. Okay, then there are certain mm, merits and the demerits of using this method, multiple or the composite cross. Okay, the first of all, the merits, or we can say the advantages. In self-pollinated crops, this method it is used when three or more monogenic characters are scattered in three or four different varieties, and these are to be combined into the single variety. Okay, in self-pollinated crop this method is used when three or four monogenic characters are scattered in three or four different varieties, and these characters are to be combined into the single variety. Okay, then generally the variety which is produced by using this method, these generally have the wider adaptations. Okay, these crosses generally have the wider adaptations. Then the limitations, or we can say the demerits of using the multiple or the composite cross in the self pollinated crop. Okay, these crosses are generally less productive. Okay, these crosses are generally less productive, and this method has the limited utility except in the high risk areas where severe disease damage occurs regularly from a highly specialized disease pathogen. Okay, this method has the limited utility except in high risk areas where severe disease damage occurs regularly from a highly specialized disease pathogen. Okay, that is all about the methods which are used in the self pollinated crop. Okay, these are the hybridization methods which are used in the self pollinated crops. Okay, there are four methods of hybridization which are used in the self pollinated crop, and these are: the first one is the pedigree method, second one is the bulk method, third one is the backcross method, and the fourth one is the multiple or the composite cross. Okay, if the question is asked in the examination as give and detailed account on the hybridization methods in the self pollinated crop, then you have to give the explanation regarding all these four methods. Or the short answer questions may be asked in the examination on the pedigree method, or the bulk method, or the mm, backcross method, or the multiple or the composite cross. Okay, and that is all about the hybridization methods which are used in the self pollinated